Okay, could you please go to the wall clap? Do you know? Let's test some knowledge here to see if you still remember your genetics course before we go to the lecture. Okay, so we will display the first question. Display. So I'm sharing my screen with you. Okay. So now you can see the code in case you are not on Brightspace. So this is the password. You can get into that. And we're gonna ask very simple 3-4 questions to see how much you remember from your genetics and how much I need to explain for you in order to understand the genetic engineering and the transgenic classes. So the first question would be, do you know what is a tissue-specific promoter?
So sorry, Professor, but at least on my end, it doesn't say which one is yes and which one is no.
Okay. I don't know what happened. I probably have wrote it but didn't save it. Let's say first, the first one is yes, 2 is no. Tissue-specific promoters. So again, 1 is yes, you know what it is, and 2, no, you don't know what it is. Okay. 2 more people in the group. Okay, so the majority do not really know what is a tissue-specific promoter. The tissue-specific promoter is a promoter that is active only in a specific tissue. In other words, your insulin promoter is only active in the pancreas. This is an example. The promoter is, in terms of the DNA, is in all our DNA, in all our cells, but it's only active in the pancreas, in beta cells in the pancreas. Okay. Next question. Again, do you know how RNA is translated to protein? 1 is yes, 2 is no. Yes, 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 yes. Too many people know how it's translated. So this means I can examine you on this and without really explaining this to you. So the majority of you know how RNA is translated into a protein, which is great. Now, what is the first amino acid in every protein? AG methionine, AG methionine, AG methionine, AG methionine. Awesome. Everybody knows that it's methionine. And the codon that encodes the methionine is the AUG. Awesome. Then, how many codons specify the amino acids of protein? So how many codons? No, 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 no. A codon is 3 base pairs. How many codons? How many in total? No, not 3. There are 3 base pairs. You are right, 3 base pairs, but how many codons in total? 64. Very close. 64 is very close. 1 codon is 1 amino acid. Very good. 1 codon is 1 amino acid. 61. 61 is the right, because there is the stop codons that do not code for amino acids. Great. So today I decided to, and before we go to genetic engineering and transgenics, I want to make sure you have the reference of what we need to know before we start transgenics. And for that, I want to make this lecture as a revision for your genetics. So for those people who don't know some of the specifics, I will give you the background. And in the next lecture, we will start constructing transgenic animals. We will mostly focus on animals, like on mice, sorry. So let me share my, stop screen, stop sharing. And let's go to the lecture here. Okay. Okay. So in order to get to the level where you can easily understand how you can make a transgenic animal, first thing you need to know is how to make the transgene. So what are the parameters that we need to take into consideration? What are the parameters that we need to take into consideration when we are generating a transgene? And then we have to find a way to deliver that DNA into the genome of all the cells in the transgenic animals. So first we have to construct the DNA, and then we have to insert it in a way to all the genome of the animal. Obviously, this will happen at the embryonic level. And then we will end up having transgenic animals, and we can identify those transgenic animals by different methods, including PCR. We know how to do PCR, but now the primers will be specified to tell us if this is a transgenic animal or not. So this might take us 2 or 3 sessions. There will be one assignment on this 2-3 sessions. I will release the assignment on Friday, tomorrow, and I will give you 3-4 weeks, I would say till December 1st, to submit that assignment. It doesn't necessarily that you need to wait till that time, but I want to give you 2-3 lectures where you can easily then perform the assignment. So up till now, we discussed many things that we will revisit today in order to start the genetic engineering processes. And next 2 lectures will be the transgenic animal generation and the types of the transgenic animals. So there's many categories we will discuss. So for today, it will be only the revisiting for the transcription and the translation process. And this is important because when we understand transcription and translation, then we can define what type of function we will address. In other words, after understanding all the processes of transcription, we can find techniques how to, for example, knock down a gene, or in other words, stop a function of a gene at the level of the RNA. This will be extensively studied. So the process of the transcription, I really want you to understand as much as possible of this. And if you have any questions, come back to me. For the translation, we will just touch it today, how the initial steps and how we get into a protein. And there will be 2 lectures about how to identify proteins, how to do Western blotting, how to do ELISA, how to do mass spectrometry, all that. It will be in 2 different lectures coming about the proteins. And this lecture today will make it easy for you later to know what is a knockout mouse, which is a mouse that lacks a particular protein, or a knock-in mouse where we have a certain DNA has been inserted into a specific site. And again, these coming in the next couple of lectures. And then we will discuss about inducible models or tissue-specific models, where, for example, you want to express a protein in a particular tissue only, or you want to knock out a protein in a particular tissue. Like I do a lot of knock-ins and knockouts in the heart. So for example, if I want to knock out a protein in the heart, I have to find a promoter that is specific to the heart, and then I have to find the gene information. So don't worry about these three. I'm just defining them now. I'm sure at the end of this course, you will be very good in identifying the different types of transgenic animals. So let's start by revisiting the transcription. So you know that transcription is the first step of the gene expression, because you are involving a copy of a specific DNA in a format of a different molecule that is RNA. So we have the double-stranded DNA. And from the assignment 2, you know that genes could be on the upper strand or 5' to 3' strand, or on the reverse strand. And when we transcribe the DNA, we generate a single-stranded RNA. Now, this process is done by RNA polymerases, and the RNA polymerases, they would read one template strand of the DNA. So if you look at this one, so this is the strand that is, this is, for example, the gene, and you are transcribing that gene by reading and adding the RNA polymerases read this template strand and translate it and transcribe it into RNA molecule. Now, if you look at the template strand, you will see that the RNA is going to be complementary, or in other words, it is identical to the non-template, with one exception, that the T is replaced with a uracil. The thymidine is replaced with a uracil. And that's how you generate an RNA. I don't want to say messenger RNA because there are many other RNAs we will discuss today, an RNA. So the RNA molecules are normally complementary and anti-parallel to the DNA strand. And as I said, the RNA transcripts have the same polarity and the base sequence of the non-template strand, with one exception, that uracil is replacing the thymidine. So this is the first initial step of gene transcription, where you are copying the information from a DNA double-stranded molecule. You pick one of the two strands, and then you transcribe that into a single RNA molecule. Okay? Let's dive a little bit more onto the steps. So what do we need in order to perform the transcription? And why transcription happen in certain areas or stretches of the DNA? And what are the requirements for the transcription? So in order to perform a transcription, we need transcription machinery, of course, or a transcription unit. And the transcription unit is defined to be a stretch of the DNA that encodes for the RNA molecule and all the necessary sequences for the process. Of course, we mentioned that we need polymerases, the RNA polymerases, which will read what is the code on the DNA and formulate what is the RNA complementary part. So I just want to quickly check. Somebody after the lecture, would you be able to post a recording? Yes, I will post a recording after the lectures. So what do we need for the transcription process? We would need a promoter, and this is now probably the third or fourth time we are visiting this word, a promoter. It's defined to be a DNA sequence that is recognized by the transcription apparatus, which is the RNA polymerases and accessory proteins that help in the transcription. It will indicate which of the two DNA strands to be transcribed or to be the template, and it would indicate the transcription direction. So if the promoter is here, then downstream of the promoter is the initiation site of transcription. And if the promoter is there, then the initiation site here, and therefore you are reading the second strand, the reverse strand. The promoter determines the transcription initiation site. So downstream of any promoter, there should be an initiation site. Okay? However, keep in mind that promoters are not transcribed. They are not part of the messenger RNA or transfer RNA or whatever RNA. They really direct the transcription initiation, but they are not part of the RNA. Then within the transcription unit, we have a stretch, which is the RNA coding region. In other words, this is the one that is copied from DNA into RNA. And finally, this process is not indefinite. So if it starts somewhere, it should end somewhere. And that's where is the transcription in termination site. So the genes are physically known where they start and where they end. And as the name indicates, it looks like a complicated name. A promoter is just a sequence. Initiation site is just a sequence. And termination sites are just a sequence. But the promoter helps to recruit the RNA polymerases and the transcription machinery. The initiation site determines where the transcription start, and the sequences of the transcription termination, it ends the transcription process. So what we end up having after the transcription is a single-stranded RNA. Since we have polarity of the DNA from the 5' to 3', and we discussed many times that addition of these nucleotides always happen from 5' to 3' end. The RNA also have the same polarity, 5' to 3' end, but now it's only a single-stranded molecule. So if we look closer, let's go on. Sorry, my computer froze. Okay. So if we look closer to the RNA transcript, if this is the RNA from the start initiation site of the transcription to the termination site of the transcription, not all of that is going to be used for generating a protein. So this is called a pre-messenger RNA, okay? Or an RNA transcript. So in the next couple of slides, I will focus mostly on the messenger RNAs because the messenger RNAs are the one that will be transcribed onto proteins. So if we look at this RNA, there should be three major regions. The first region is the 5' untranslated region. In other words, it will be transcribed from DNA into RNA, but it will never be translated into a protein. And then there's a start and an end codons you can see here. In between, there is the protein coding region. And then there is also a 3' end that is not translated to the protein. So in other words, the protein coding part of a messenger RNA is much smaller than the pre-messenger RNA. Okay? Now, how do we get from a messenger RNA, that the pre-messenger RNA, into a mature messenger RNA? So we would go first to the RNA processing steps. So there are 4 steps that you would need to know, that in eukaryotes, the RNA molecules must be processed after transcription. And this processing are capping of the 5' end, adding the polyadenine to the 3' end, cleavage of a part of the 3' end, and then splicing. And we visited splicing multiple times, but I will go one more time today. So these are the sites. So first, well, the order could be that cleavage first, and then, sorry, cleavage first. They are listed here, but not in the order. So the cleavage of the 3' end, then the polyadenylation, where you add A at the end of the messenger RNA, and then the capping, which is addition of this methylguanine. And I don't really expect you to know this structure or the name. I want you to know that there is a capping that's taking place at the 5' end. I don't want you to memorize this. Okay, then as we discussed in the previous lecture, that the biodiversity partially comes from the splicing. And the splicing is, in principle, if you have a gene that contains a promoter here, and then there's exon 1, exon 2, exon 3, and there's 2 introns. When you transcribe this gene into the pre-messenger RNA, you will generate a 5' end that will be capped. You will generate all the sequence, including the introns and the exons. So you have exon 1, intron 1, exon 2, and all the way to exon 3. Now this area here, or the site that is not needed, can be cleaved. So this is, I showed you on the previous slide. Polyadenylated, so there's a lot of A's so that it's protected from degradation. But what you can see here, that we can generate 2 different messenger RNA molecules from the same pre-messenger RNA. So this pre-messenger RNA is being spliced. Well, after polyadenylation, it's being spliced, where exon 1, 2, and 3 could generate this mature messenger RNA, and therefore it will generate a different sequence of a protein. And on the other side, if you take exon 1 and exon 3, this will generate another different type of protein because different sequence. Therefore, the RNA splicing is one of the essential processes that could lead to more biodiversity because we are using the same DNA molecule to generate the same pre-messenger RNA molecule, but then two different mature messenger RNAs, and in other words, two different mature proteins. Okay? Questions for now? No questions. I don't know why when I'm using this pointer, it doesn't allow me to go from slide to slide. So this slide summarizes the transcription from the beginning till the end. So you have a promoter. Downstream of that promoter, there is a gene. We know what initiation site of the transcription. We know the end of the transcription. This particular gene within the RNA coding region, there's one exon, three exons and two introns. We transcribe this from the initiation site to the transcription end site. Then there's a cleavage of the 3' end. There's the addition of the poly A tail. There's the capping that took place at the 5' end. And then the alternative splicing where introns are removed. And then we end up having a mature molecule of the messenger RNA. Great. So at this point, we have a mature messenger RNA, and now we want to translate this into a protein. So before going to the process of the translation, I just want to mention that there would be more than one class of RNA in cells based on which cell type you are studying. So prokaryotes versus eukaryotes like bacteria and yeast, bacteria versus human, there's different classes of RNAs that are expressed or not. In these sessions, we would like to know information about the messenger RNA because that's how we can manipulate gene function. We will discuss a little bit about the transfer RNA because that's how the translation process takes place. And we would like to also discuss the microRNA and the small interfering RNA because that's how we knock down genes. We can down-regulate the expression of a gene by simply using the microRNA or the small interfering RNA. So one of the examples that probably you already know is using the messenger RNA in vaccine has never been done before, except with the COVID-19 vaccine. And you heard a lot of conspiracy theories about the COVID-19 vaccine. I will have a slide just describing this quickly. And then when we discuss proteins, we will revisit this slide again. So in summary, DNA is transcribed into many RNA. Each one of these has its different function. And the translation of some of these RNA will lead to the protein. Not all the RNA categories lead to the protein synthesis. The messenger RNAs are the one that are translated to the protein. All the rest play regulatory roles in the transcription process. Okay, so let's discuss a few words about the messenger RNA vaccine. And I don't know how much you know about how vaccines are generated. But in order to generate a vaccine, you have to identify a specific protein that identifies a particular virus. In this condition here, the spike protein would identify COVID virus 19 compared to any other viruses. In other words, this spike protein should pick only COVID-19 out of any other viruses that you could imagine. So previous methods of generating a vaccine was to get a piece of a protein expressed, and that piece of a protein causes an immune response, and then that immune response generates antibodies against the virus, and that's how you acquire immunity. Now, probably what I'm saying is very difficult for some of you guys, or probably you have never thought or heard about how vaccines are made. If it's the case, please let me know right now. Like, unmute yourself and tell me I have never heard about this process before, so that in the next few lectures, I could include how vaccines are made for you. Now, the only difference that was introduced with the COVID-19 virus is instead of using the protein as a part to make an antibody, researchers took a messenger RNA that encodes for that protein. So in principle, if I go back, they went into the spike protein and they said, oh, this is the messenger RNA that codes for COVID-19. And therefore, if I want to make that part of the virus, which is the spike protein here, all I need to do is just give the cell the messenger RNA. And then my muscle cells is gonna translate that messenger RNA into a protein. And since this protein is not self, is not part of our body, our immune response is gonna detect it, and it will make an antibody against it. And this antibody is the antibody that gives you the immunity. Okay? Again, probably this is difficult. You have to know how the vaccines are made in general. The only thing I want you to know is that messenger RNA has been used to generate a vaccine. And it's the first time that has been used in a human. And there were a lot of questions about that. So let's continue the revision process. So we reached here that we generated a mature messenger RNA. So what do we do with this messenger RNA? How do we read it? How do we code the information in the messenger RNA? So to decode the information on the messenger RNA, we would need the protein synthesis machinery in order to perform the translation. And in this process, we will read the codes of the messenger RNA, and we will transfer that or translate that into a polypeptide, which is a group of amino acids. So we would need first the mature messenger RNA. We would need a transfer RNA, and I will describe this in a second, and the machinery, which is the ribosomes. And of course, we will need the individual amino acids. So the process of the synthesis with the machinery, it includes first the large and the small subunits of the ribosomes. These are normally not together. When the process of translation starts, these assemble together. We would need the messenger RNA and then the transfer RNA charged with amino acids. And you can imagine that you have at least 20 different combinations of that, because each transfer RNA should hold one amino acid. As an example, this transfer RNA is charged with serine, the protein, the amino acid serine, okay? And what dictates which amino acid is being charged to the transfer RNA is the anti-codon that the transfer RNA has. So in other words, if I have a messenger RNA, and this is one of the codons on the messenger RNA, then in order to translate the messenger RNA into a protein or an amino acid, then I would need a transfer RNA that holds on one side the serine or the amino acid, and on the other side, the anticodon that is complementary to the messenger RNA codon. And you can imagine for the other 19 amino acids, there should be another transfer RNA carrying different codons. Okay? So that's how we can decode the information on the messenger RNA, which is a nucleic acid, into a amino acid, which is a protein, so totally different molecules. Okay, so going forward to discuss the question I asked you, there are information on the messenger RNA, but the sequence of the messenger RNA is based on a uracil, cytosine, adenosine, and guanine. So in principle, if you take these 4 by 4, you have 16 different combinations, right? But since there were a lot of research to understand how many nucleotides are necessary to make a single amino acid, how many nucleotides you need to make a single amino acid, there were a lot of theories, and then we ended up knowing that a codon of a messenger RNA is only 3 bases. And therefore, now if you take the 4 by 4, and then you add the third position, which is another 4, that's where it comes the 64 genetic code in the 64 genetic codons. So that's where the number comes from, because you have 3 different, or 4 power 3, in other words. So each of the messenger RNA codons, which is a 3 bases, specifies a unique amino acid. And these are presented here in the first 3 letters, like phenylalanine, leucine, proline, threonine. I don't want you to know which codon encodes for which amino acid. This is not a type of question that I would ask, okay? Now, given that the initiation of the translation always happens with methionine, and I'm so happy that you know it's methionine, your answer was 100% correct. There are 3 codons that would stop the process of the translation. So whenever you reach a UAA or UAG or UGA on a messenger RNA, the translation process stops. So these are called termination of translation or termination codons. Now, the individual transfer RNA should carry the specific amino acids, as I said before, and I mentioned that one of the codons that could code for serine, it's the UCC. Therefore, if you are reading on the messenger RNA and you reach a UCC, the transfer RNA is going to add a serine to that. Now, bear in mind that some amino acids are encoded by more than one codon. So if there was a UCU on the sequence, if there was a UCU here, then we could add another serine. So in the translation process, a transfer RNA carrying an amino acid specified by the codon on the messenger RNA enters the ribosome and donates the amino acid to the growing polypeptide chain. So as the process of translation continue, we will read more on the sequences, and we will be donating our, the transfer RNA will be donating this, and the peptide will be generated. So if we look at the process from A to Z in terms of the translation, so we discussed that a gene is transcribed, we have the messenger RNA, and now this messenger RNA the translation process should start with the AUG, which is the methionine, the two subunits of the ribosomes, the large and small, and the first anticodon, which is complementary to the AUG, is going to carry the methionine. It binds, and then the process of translation starts. When the first three codons are read, ribosomes are going to move to the next. Each codon is being added, and you can see here we have 1, 2, 3, 4, 5, 6, 7 amino acids. And now you are already at the 21 base pairs. So 1, 2, 3, 4, 5, 6, and 7 amino acids. And within the initiation process, the assembly take place, and the elongation process is simply that you are continuing to read till you reach the stop codon. Once you reach the stop codon, then there will be the release of this transcription machinery, translation machinery. And the amino acids that has been linked together, they will form this peptide, which is the protein. Therefore, through the process of translation, the amino acids are linked together in a specified manner based on the RNA sequence. Okay? And that will generate a new class of molecule, which is the protein. Now we are talking about messenger RNA, which is a nucleic acid that is translated to a protein. So what are the proteins? And from the biochemistry, I'm assuming you should know this. But again, I want to really go back from the basics with you, because in the next couple of lectures, it would be much easier that we have this background. So what are the amino acids? The amino acids, these are acidic molecules that has an amine group and a carboxyl group. Also, it has this hydrogen bond. So you know that when you have hydrogen, you can add nothing here because there cannot be any chemical reaction that take place on hydrogen. Plus, we have the radical group, which is a side chain, and this radical group is different from one amino acid to another. This is what makes methionine is different from leucine. Depends on what you add on this group. Okay. Now, when we add amino acids in peptides, so if we go back just one step here, so we have the first amino acid, the second, the third, the fourth. Yes, the information comes from the messenger RNA. We read a codon of 3 base pairs, and then another codon of 3 base pairs, and another codon of 3 base pairs. And then we are adding the first amino acid for the first codon, second amino acid for the second codon. And so there is a form of polarity that the messenger RNA dictates in the protein. So it's followed by the translation process. However, you have to know that amino acids are also, the peptides are also polar in a sense that the amino acids are joined together by peptide bonds. And these peptide bonds are added to the carboxyl group. In other words, every protein has an N-terminus where you have the amino group, and the peptides will be added here, as you can see down. So yeah, we have one amino acid and another amino acid. We add it to the carboxyl group, the second peptide amino acids. So we formed a peptide of 2 amino acids. The third peptide will be added also to this carboxyl group, and therefore any protein should have an N-terminus and a C-terminus. Okay? Now, proteins are the functional units of our cells. Everything you see in your body is a protein. Your skin is made of proteins. Your blood is all proteins. The hair is proteins. Like everything you see in terms of function is practically proteins. So the proteins can be enzymes, they can be catalysts for a lot of activities. They could be part of your structural component, like scaffolding, making membranes, making filaments, making bone, making hair. The differences is basically in the sequence of the peptides. Your antibodies are proteins. The proteins for transport of a chemical or something, that like everything you do is any function in the body is basically performed by specific proteins. Even the process of the transcription and the translation is performed by proteins, because it's RNA polymerase that transcribe the DNA, and DNA polymerase is used for transcription. And the translation machinery that we just described here is in principle composed of proteins. So it's important for you to know that everything we are studying right now in this whole course will end up in one particular aspect, where how do we utilize proteins and how we can manipulate proteins. And when you say, I want to generate a transgenic animal, you will end up affecting a function of a protein, either overexpressing a protein or downregulating a protein. Okay? So the proteins are generated from the messenger RNA sequences. And as we said, it's based on the RNA sequence, which dictates the N to C terminus of an amino acid or of a protein or a peptide. And therefore, the sequence of the different amino acids is called the primary structure of a protein. The sequence also dictates how the amino acids fold, because you know the amino acids are charged. They might like each other or they might repel each other, and therefore that causes the proteins to make secondary structures. So the interactions between the amino acids could make the protein fold. I don't want to go to the structures, what is alpha helix or beta, or I'm not going to this detail here. There are many structures. Now, the proteins which consist of more than one polypeptide could have the quaternary structure. So this is something you have to know, that the primary structure and the secondary structure and the tertiary structure are mainly through the sequence of the amino acids, while the quaternary structure, it means that you have more than one peptide. So you have one peptide, another peptide that together make a protein, and that's what we call the quaternary structure. So an example of that is hemoglobin. You all know that hemoglobin, whenever you do a blood test, there are multiple chains. You have the alpha and the beta chains of the hemoglobin, and there are the iron group or the heme. That's how the name comes. Now, if this is important to know, that the primary, secondary, and tertiary structure or the folding of the protein is part of the protein function. It is important for the protein function. Because if you lose the structure, you lose the function. The best example I can give you, when you fry your eggs, and you want to have a breakfast in the morning, and you have the egg, it's full of proteins. If you fry those eggs, what you do is you unfold these proteins. That's why it totally changes. So that being said, we would like to discuss a couple more categories of the RNA, which are basically the microRNA and the small interfering RNA. So let me go back a couple of slides just to link things easier. We said that whenever we have a gene under control of a promoter, we end up having an RNA. In this condition, this is a messenger RNA. What this slide is telling you that there are multiple categories of RNAs. We discussed what the transfer RNA does, and we will discuss how we could generate a microRNA and where small interfering RNA come from and how do they function. The microRNA and the small interfering RNA are basically used for gene silencing. So if you want to block the function of a gene, you would need, not you would need, in certain aspects, we know that microRNA and small interfering RNA can perform this function. And since we are dealing with research approaches, you can imagine that microRNAs and small interfering RNAs are used heavily in research because they are the tools that we can use to knock down genes or downregulate genes. So let me go to that slide back. And let's discuss how small interfering RNA and microRNA are generated. So you remember that RNAs are supposed to be single-stranded. But if a specific RNA has been generated from a gene where within that gene there are some inverted repeats, and if you're not familiar with the term inverted repeats, these two sequences here, they could make the RNA complementary to each other. In other words, if you look here, the UCAGG is gonna be binding with the AGUCC to make this fold. So U and the A, then the C with the G, all the way to the end, will form this loop of RNA. And normally RNAs are single-stranded, but when they are double-stranded, then they are recognized by certain enzymes called dicer. Okay? Let me go to the next part of the story. So we continue here. When the dicer works, it will cleave the RNA into smaller double-stranded pieces. So it will produce smaller double-stranded pieces. Those pieces or nucleotides, they are between 19 to 23 nucleotides in general. And they are double-stranded RNA molecules. They will be bound by another protein called RISC, which is RNA-induced silencing complex. Then the RISC, there is a selection process. It will take only one of the two double-stranded RNA. It will release one of the two, and that sequence will be complementary to a gene that once it binds to that gene, it will inhibit the translation process. Now that's for the microRNA. So microRNAs, we know a lot of microRNAs, and they are genes, but instead of generating a messenger RNA, they just generate these small stretches of RNA that is not translated, and then that is processed to generate a molecule in combination with the risk to downregulate particular genes. Now, of course, there are more details. We will describe this, how we use it. I'm just introducing the theory. Now, the small interfering RNA is not very clear the source of the small interfering RNA. It could be probably through the infections we got with the viruses. But what happens is that the double-stranded RNA is generated. And whenever this double-stranded RNA is generated, it will be recognized by the dicer, the same process that we discussed with the microRNA. So the only difference between microRNA and small interfering RNA is the source of the double-stranded RNA. And then the processing of the small interfering RNA is exactly the same as the microRNA, where it will be degraded into smaller 19 to 23 nucleotides of double-stranded RNA that are recognized by the protein and form together what we call a RISC. And obviously, you choose one of the two of these RNA templates that you would direct the gene degradation. So the source of small interfering RNA compared to the microRNA is not clear. And finally, the microRNA would block the translation process. It inhibits the process of translation because if the translation machinery comes, it's not going to be able to bind to the gene. While small interfering RNA, it causes messenger RNA degradation. It will destroy the messenger RNA. And in both situations, what happens is you don't produce a protein out of that gene. Now, how do we choose which target we get is very important, because when you have the microRNA that is bound to the protein, this has a particular sequence. Therefore, it will recognize genes that has complementary to that particular sequence. And the same thing here. When you have the degradation, the dicing of the siRNA, then you will get these 19 to 23 base pairs into the RISC, and that will, based on the sequence of that messenger RNA, the 19 to 23, it will recognize specific genes, not any other, like not any random gene. And then it will degrade the messenger RNA of that gene. So in the future, few lectures, I will give you a concrete example how this is being used. And I will show you how we generate in the lab. Exactly like you designed a primer, you can design oligos, 19 to 23 base pairs. All I need is just the gene of interest. Say this is, for example, albumin gene or insulin, which insulin is not going to be a good example. Let's say albumin. And you want to degrade or block the generation of albumin. Then you will generate 19 to 23 base pairs complementary to albumin, and you will transfect it to the cells as we described before in the transfection process. And this will bind to any messenger RNA generated from albumin, and it will degrade. So there will be no more albumin generated in the cells. So that's how we can achieve a gene knockdown. Okay. Now let's revisit some of the, well, let me stop here and take some of your questions better. So I will stop and take some of your questions, if you have any. If there is anything not clear, you can unmute yourself or you can just start writing in the chat. Any questions? No questions? Are you there? Okay. So either this is very easy for you or it's very difficult. I don't understand you. Anyways, you don't have any questions. I will go forward then. So must review, yes. Because in the next lectures, I don't want anyone to say, uh, the assignment, Maggie, is gonna be that I will give you a paper and I want you to identify what type of transgenic animals are those. Like, is this a tissue-specific animal? Is this an overexpression? Is this a knockdown? Okay. Okay, sounds good. So let me move forward with the examples. There's only 3 more slides for this lecture. And I hope you guys digested very well, even though you are telling me now that it's mostly review. That's great, but you have to put this into practice. You have to start putting this into practice. So remember when we were discussing transfection processes, and this was one of your exam questions, that what are the 4 major aspects you should have in a plasmid? One of those was a promoter. So I hope now it makes more sense for you why we have these promoters on the plasmids. Because the promoter, based on this old slide, a promoter to drive the expression of the gene of interest. And promoters can have various strengths. So could be constitutive promoter, means it's an active under all circumstances. While inducible promoters, and that's what we will get in the future, in the inducible transgenics, becomes active only when a stimulus or can be modulated to switch on and off. So the inducible promoter, we decide when we turn it on and when we turn it off. So now it makes sense for you that if you want to express a gene of interest, you have to select the right promoter. And that promoter, in vitro, when we do research, we mostly use promoters that are active from viruses. This slide also was from previous lecture where we discussed that when we do recombinant, one of the applications of the recombinant DNA technology is to produce proteins such as insulin. Now, in order to, if I ask you now, are you able to put a plan for generating insulin in vitro? What would you need? Like what exactly you should do in order to be able to overexpress or generate insulin in vitro? So in principle, in order to overexpress or express, not only overexpress, a gene, you would need a promoter. And in this situation, this is a cytomegalovirus promoter. It's a virus that affects us, but its promoter is widely used in research because it's active all the time. And it's being used in transgenes expression in mammalian cells. Any of the labs you would go, you will get some sort of research that's happening with the plasmids that they have CMB promoter. Then, from the assignment, you know that if you want to express a gene, you don't have to express all the messenger RNA. You need the coding DNA, which is the mature messenger RNA. And if I go back in my slides up to here, when you want to generate a transgenic animal, You need the promoter and then the coding sequence, which is complementary to the mature messenger RNA. Therefore, in the next lecture, we will generate the first transgenic animal, which is using a simple promoter and a coding sequence. For example, if we would use the gene insulin as an example, we have to go and find the coding sequence from the first ATG till the stop codon of the insulin gene. This is an example of transgenic animals. So transgenesis is one of the tools that has been described and being propagated since 1980s. The term transgenesis is in principle adding foreign DNA or genetic material to an organism's genome. This means it has to be retained in the genome throughout the life of the animal. And nowadays, there are tons of transgenic animals that has been produced, and they are really a major tool for the health biosciences. You could, for example, here, we are trying to visualize how ubiquitin promoter works. So we could use transgenic animals to label proteins and visualize their localization and trafficking. We can study the function. We can make a gain of function by expressing or loss of function by deleting a protein. And the example you see here is the green fluorescent protein. We use a ubiquitin promoter. So this is a transgenic animal, transgenic mice, express green fluorescent protein directed by the promoter, which is the human ubiquitin promoter C. There are multiple types of ubiquitin, so human ubiquitin C promoter. In other words, whenever the protein, the promoter, the ubiquitin promoter turns on, it will generate a mature messenger RNA and a mature protein that is green fluorescent. Every time the promoter is on, you will generate a green fluorescent protein. And therefore, you would know whatever the ubiquitin promoter is on, where is the generation of that protein. And since the ubiquitin is expressed everywhere, if you take this mouse and you put it under microscope, you can see that all the body expresses GFP or green fluorescent protein. So this is a very simple example how we use transgenics. Okay, now for the next couple of slides, I want you to recall one important notion about meiosis and mitosis. Probably you already know this, and I can see that you guys have good background, that when you have mitosis, most of the time you end up having a duplication of the cell with the exact amount of genetic material. While when we have the meiosis process, we end up having 4 cells. Each of them has half of the amount of the genetic material. Now, mitosis happens in somatic cells, so every cell that is not a gamete, and meiosis happens only in sex cells or gametes. So in other words, mitosis end up producing fully functional 2 copies of the same cell. Like this happens in your skin, in your intestine, and everywhere. While the meiosis happens only in the sperm and in the egg. And therefore, you produce many In condition of male, you have 4 functional sperms. In condition of female, you have only 1 egg that is being produced monthly. And that's why during the menstrual cycle, the woman gets either pregnant or the menstrual cycle happened to release that egg. Why this is important? Because when we deal with transgenics, we have to know at which step we have to utilize the gamete and the egg. So looking from this point of view, we can say that meiosis generates a gamete that has half the amount of the genetic material. And fertilization, when sperm and egg meet together, could restore the genetic material. And that's how the life continues. One of the aspects of the genetic diseases is that if you have more or less of the genetic information, that can cause a disease. And that's why gametes can never be viable unless the fertilization takes place. That's why, as I said, menstrual cycle happen because the egg has, in a female, has half of the amount of the genetic material. And since no fertilization happens, it has to be removed. If defertilization happens, then the implementation and the embryo start to propagate. Now, of course, there's too many other things that regulate this, like hormones and such, but we will take this into consideration when we generate transgenic animals. Okay, so with this would be my last slide. If you have questions, let me know. And expect tomorrow a release of the assignment. You cannot start working on the assignment before the next lecture because you have to understand the difference between the transgenic animals before you can go to the paper and dig what are the transgenic animals in that paper. Okay, thank you very much. Okay. No questions? Anybody wants to stay to discuss? Anybody have any concerns? All good. By the way, I forgot to tell you, there will be 1 bonus point that will be added to your midterms because one of the questions was not displaying correctly, and I will add the 1 bonus point to everybody.
Hi, Professor. Can you hear me?
Just give me 1 second, please.